Assalamualaikum, dear students. Today we will discuss the remaining lectures of protein metabolism.
Bismillah ar-Rahmanir Rahim. Students, the next topic is catabolism of amino acids. The pathway by which standard amino acids are catabolized are conveniently organized as into different 6 ways. In first way, amino acids that form oxaloacetate. The second way, in which amino acids that form alpha ketoglutarate via glutamate. In third way, amino acids that form pyruvate. The fourth way, in which amino acids that form fumarate. Fifth class of the amino acids that form by different metabolic pathways with the action of different types of enzymes, succinyl coenzyme A. And in the sixth way, in which amino acids by the action of different biological catalysts to form acetyl coenzyme A. Now we will discuss each way separately. The first way, in which amino acids that form oxaloacetate, is asparagine. Asparagine is an amino acid which is hydrolyzed by asparaginase and liberating ammonium and aspartate. Aspartate loses its amino group by transamination to form oxaloacetate. The reaction is presented asparagine by the action of asparaginase converted into aspartate, and aspartate in the presence of aminotransferase enzyme to form oxaloacetate. The second way, in which 4 amino acids, that is glutamine, choline, arginine, and histidine, form alpha ketoglutarate via glutamate. Glutamine amino acid is hydrolyzed to form glutamate and ammonium by the enzyme glutaminase, and glutamate is converted to alpha ketoglutarate by transamination or through oxidative deamination. And when the oxidative deamination will be, then the enzyme will be glutamate dehydrohalogenase. Choline form alpha ketoglutarate in such a way that amino acid is oxidized to glutamate, and glutamate is transmitted or oxidatively deaminated to form alpha ketoglutarate. Arginine, this amino acid is hydrolyzed by arginase to produce ornithine. Ornithine is subsequently converted to alpha ketoglutarate with glutamate semi-aldehyde as an intermediate. Histidine, this amino acid is oxidatively deaminated by histidase and finally formed alpha ketoglutarate. Collectively, the reaction of glutamine, choline, arginine, and histidine in conversion of glutamate, and finally to alpha ketoglutarate is presented on this slide. In third way, some amino acids like alanine, serine, glycine, cysteine, and threonine to form pyruvate by the action of different enzymes. Alanine, this amino acid loses its amino group by transamination to form pyruvate. Similarly, serine can be converted to glycine and are intermediate compound, and finally it converted into pyruvate by serine dehydrohalogenase. Glycine, this amino acid can be converted to serine by reversible addition of a methylene group from intermediate acid are oxidized to carbon dioxide and ammonium. Cysteine, the amino acid, undergoes desulfurization to yield pyruvate. You know, cysteine is a sulfur-containing amino acid and it removes its sulfur to form pyruvate. Threonine, this amino acid is converted to pyruvate in most organisms but is minor pathway in humans for its conversion, and collectively the reaction of serine, glycine, threonine, cysteine, and alanine is presented as is into conversion of pyruvate by the action of different type of enzymes. In fourth way, some amino acids that form fumate, phenylalanine and tyrosine are those amino acids which can contain phenyl ring in their structure. Hydroxylation of phenylalanine produces tyrosine. This reaction is catalyzed by tetrahydrobioprotein requiring phenylalanine hydroxylase for its further activation and initiates the catabolism of phenylalanine. Thus, the metabolism of phenylalanine and tyrosine merge, leading ultimately to the formation of fumate and acetoacetate. Phenylalanine and tyrosine are therefore both glucogenic and ketogenic amino acids. Phenylalanine converted into tyrosine and tyrosine to form fumate and acetoacetate. In the fifth step, way, amino acids that form succinyl coenzyme A, methionine, valine, and isoleucine are those amino acids which convert it into succinyl coenzyme A by different type of biological catalyst. For valine and isoleucine, vitamin B12 and biotin are required as a coenzymes, which is converted into succinyl coenzyme A, and succinyl coenzyme A finally enter into the tricarboxylic acid cycle. In sixth way, there are some amino acids like leucine, isoleucine, lysine, and tryptophan. They form acetyl coenzyme A, and acetyl coenzyme A further enter into different metabolic pathways. And these amino acids converted into acetyl coenzyme A in the presence of different enzymes. Ultimately, the standard that are 20 amino acids are converted into their intermediate compounds, which are further catabolized and metabolized by different cycles occur inside the cell. Thank you very much.